Good morning, dear student. In today's lecture, we are very much interested in the new chapter of our course CH508 Chemistry of Biomolecules, that is enzymes. So, what is this enzymes? What are the roles of these enzymes? So, we all discuss in this lecture. So, let us discuss some important points or important characteristics regarding to these enzymes. So, these enzymes, they are biocatalyst. Okay, so we know what is catalyst, what is the role of catalyst. Catalyst, it is any substance which increases the rate of chemical reaction. Okay, so this is your biocatalyst, means it increases the rate of different biological processes, metabolic processes. Okay, so this biocatalyst, these enzymes are act as a biocatalyst which increases the rate of different metabolic processes by lowering the activation energy. So same as a catalyst, it is used in biological process or different metabolic process in the cells. Okay, metabolic process in the cells, and it is synthesized by the living cells. So the first point is that the biocatalyst enzymes are the biocatalyst, it is synthesized by the living cells. Second important point is that like other catalyst which is used in chemical reactions, enzymes increases the rate of the reaction, any metabolic process, it increases the rate of metabolic process by lowering the activation energy, by lowering the activation energy. Third important point is that like your catalyst used in chemical reaction, this biocatalyst, that is your enzymes, are not consumed in the reaction and not alter the equilibrium, not alter the equilibrium, that is equilibrium is not shifted from reactant side or product side. So it increases only the rate of reaction, it increases only the rate of reaction, it is not consuming the reaction and not alter the chemical, not alter the equilibrium. Okay, one more important point regarding to these enzymes, that is they are specific in their action, they are specific in their action. Again, means they are specific in their action, so different enzymes use different product with same substrate, therefore they are act as a specific in their action. Okay, the next important characteristic is that they lost their activity. Okay, they lost their activity when subjected to the heat, strong base, strong acid, or organic solvents. Okay, so these are the some important points regarding to these enzymes. So they are act as a biocatalyst synthesized by the living cells, it increases the rate of reaction by lowering the activation energy. Again, third important point is that it is not consumed in the reaction and not alter the equilibrium. They are specific in their action and they lose their activity when subjected to the heat, strong base, strong acid, or organic solvent. So let us discuss some classification of these enzymes. Let us discuss the classification of this enzyme. The enzymes are sometimes are broadly classified. These enzymes are broadly classified into two types. Okay, one is intracellular intracellular enzymes, and second one is extracellular enzymes, extracellular enzymes. Okay, one is your intracellular and second one is your extracellular enzymes. Okay, if you consider these intracellular enzymes, they are functional within the cells. Okay, so these intracellular enzymes, they are functional within the cell, within cell, within the cells, they are functional within the cells. So this is the important points regarding to this intracellular and extracellular enzymes. Intracellular enzymes, they are functional within the cell where they are synthesized, while these extracellular enzymes, these are the enzymes, or these enzymes are active outside the cells. These extracellular enzymes, they are active outside, they are active outside the cell. So you should keep in mind that is intracellular enzymes, they are functional within the cells, while extracellular enzymes, they are active outside the cells. Okay, again, if you consider the International Union of Biochemistry, it is known as capital IBU. So what is this IBU? IBU, so it stands for International Union of Biochemistry and Molecular Biology. International Union of Biochemistry International Union of Biochemistry, and along with this International Union of Biochemistry, Molecular Biology. Molecular Biology. So both these two, that is International Union of Biochemistry and Molecular Biology, have developed some basic principles for classification of the nomenclature of enzymes. So they develop both these two, that is IBU, International Union of Biochemistry, and Molecular Biology. They develop, okay, they develop they develop some principles, develop some principles they develop some principles for classification and nomenclature of enzymes. Develop some principle for classification and nomenclature and nomenclature and nomenclature. So you should keep in mind this IBU, International Union of Biochemistry and Molecular Biology, develop some principles for classifications and nomenclature of these enzymes. Okay, and according to them, the enzymes are classified into 6 major classes. Enzymes are classified into 6 major classes. So these enzymes are classified into 6 major classes. The first column stands for enzymes, then the reaction which is carried out by these enzymes, or which increases the rate of this reaction, and the examples is there. Okay, so this is important point. Okay, so the first enzyme is your oxido reductase. Oxido reductase. So the name itself indicate what is its function or where it is used. It is used in oxidation reduction reaction. It is used in oxidation reduction reaction. Oxido reductase. So oxidation reduction reaction. Oxido reductase. It is used in oxidation reduction reaction, and the example is alcohol dehydrogenase. Alcohol dehydrogenase. Alcohol dehydrogenase. Okay, so at least up to this you should just understand what are the different enzymes, where they used, which kind of reaction, or what kind of reaction, in which reaction they are involved, and how they increases the rate of reaction, and at least you should know the examples. Okay, so we are not goes into very much details about this alcohol dehydrogenase. In later on we discuss all of this. Okay, so at least you should know the name of the enzymes, where it is used, which kind of reaction it is performed, or which increases the rate of the reaction, and the examples. The first one is oxido reductase, which is used in oxidation reduction reaction. So it increases the rate of this oxidation reduction reaction, and the example is alcohol dehydrogenase. Second one is your transferase. So what is this transferase? What is the which kind of reaction they perform, or which kind of reaction are carried out with the help of this transferase? That is the group transfer. The group transfer. So transfer of group from one molecule to the another. So this is the general reaction, that is AX plus B, and there is a transfer of group X from this A to B. So we get this. Okay, so this is your group transfer reaction, which is carried out with the help of this transferase enzymes. Okay, and the example of this hexokinase, the example of this transferase enzyme, it is your hexokinase. So different examples are given, so we just consider only one example here. So the second one is transferase. Its function is transfer of group from one molecule to the another molecule, and example is hexokinase. The third one is that is hydrolysis. Hydrolysis, the name itself indicates its function, that is it is used in hydrolysis reaction. So we are very much familiar with this hydrolysis reaction, that is the reaction of any molecule with water. Generally we consider the water, then the cleavage of bond between the AB molecule by the addition of water molecule, and we get here H plus BOH. So it is called hydrolysis reaction, and this hydrolysis, it is involved in this hydrolysis reaction, and the examples of this hydrolysis enzymes, that is your lipids. The very famous example of this hydrolysis, that is your lipids. So its function is hydrolysis. Then the next one, that is your fourth one, that is your lysis. Okay, so lysis. Okay. So this lysis, it is involved in addition elimination reaction. It involved in addition elimination reaction. It involved in addition elimination reaction. Okay, and its example is aldolase. Its example is aldolase. It is involved in addition elimination reaction. Addition elimination reaction means this AB is there plus XY is there, so we get the formation of AX BY. So they involve in addition elimination reaction. So at least you should understand or keep in mind the name of the enzymes, where it is used, and the examples. Okay, so oxido reductase, transferase, hydrolysis, lysis. Okay, one more is there, 1, 5 and 6 is also there. Okay, so we'll discuss this. The fifth one is, that is your isomerase. The fifth one is your isomerization reaction. So interconversion of isomers. Interconversion of isomer, just like cis trans isomerism. Cis is converted into trans, and trans is converted into cis in organic chemistry. So this is the enzyme, that is isomerase. It converts one isomer into another isomers. That is A is converted into A star, and A star is converted into again A. And the example of this isomerase is your triose phosphate isomerase. So there are different examples, we consider only one, triose phosphate isomerase. triose phosphate isomerase, and the sixth one is that your ligases or ligases. Okay, so it is used in the process that is condensation. It is used in condensation reaction, that is A plus B gives AB. Okay, this is your condensation reaction, and generally in this case uh, the example of this ligages, that is ATP, is converted into ADP. Again, the one example is given, that is ATP, adenosine triphosphate, it is converted into adenosine diphosphate. This is your condensation reaction. Okay, and the example is your glutamine synthesis, glutamine synthesis. Okay, so this is all about the classification of enzymes. In the next lecture we will discuss the active site of these enzymes and the specificity of this enzyme. Thank you.